we will look at the contributions of Lashley and Hebb in neuropsychology. Neuropsychology is related to cognitive psychology. Neuropsychology is the scientific study of the relationship between the nervous system and both cognition and behavior, between the nervous system and cognition, between the nervous system and behavior, both of those things. For instance, neuropsychologists might ask questions like, what does the brain do when we speak? What does the brain do when we run a marathon? What does the nervous system do when we study for an exam? What does the nervous system do when we're asleep? All of these questions might be of interest to them. This subfield of psychology is also known as physiological psychology or neuroscience. When I was an undergraduate student, I worked with 2 neuroscientists and learned how to use some pretty cool technology. The term neuropsychology was first used in the 1910s and then popularized by Lashley in the 1930s and Hebb in the 1940s. During the time that neuropsychology, cognitive psychology, and the other types of psychology were just beginning to emerge, there were several people outside of the field of psychology who were developing, creating, inventing new types of technology that could be used to measure the activity of the nervous system of the brain. In 1924, the electroencephalogram, the EEG, was invented. That same year, it was discovered that the 2 hemispheres of the brain communicate through the corpus callosum, which is inside but between the 2. In 1936, Freeman conducted the first human lobotomy. We learned about him in last week's lecture. In 1953, rapid eye movement was discovered, the notion that during different stages of sleep, the brain activities associated with that stage vary. Then it was discovered in 1962 that the 2 brain hemispheres are responsible for different functions of the human. In 1972, magnetic resonance imaging, the MRI, was discovered. Later that year, the CT scan, the computed tomography scan, was invented. The year after that, the PET scan, positron emission tomography, was invented. Several decades later, and of course, there were many other types of technology being created, updated during this time period, but I wanted to mention that in the early 90s, the functional MRI is commonly used today in neuroscience and neuropsychology. On the next couple of slides, I want to show you what the output looks like for some of these different types of technology. Here are several examples of brain wave activity that we might see if we used the EEG. This is an example of what the output looks like for an MRI. Here is an example of what is created by conducting a CT scan. And finally, we have the output from a PET scan. The areas in red are where the most activity is occurring. The areas in purple and blue and black are the areas where there is the least amount of activity. So you can see the difference between hearing words, seeing words, speaking words, generating words. These different actions require different parts of the brain. A PET scan shows us which areas are activated during specific activities. I'll say it one more time, neuropsychology developed along with the technology. These different techniques that I've shown you on the last few slides are still used today in some form, especially in neuroscience research. Again, as an undergraduate student, I used the EEG with more than 100 different participants. You place a cap on the head, it has electrodes, they have to stay wet, so you're always putting stuff in people's head, it goes in their eyes, and you got to dab it. It was a very time-intensive study to conduct using the EEG cap, but it was really interesting to get to see how different people's brains react to different kinds of stimuli with such a simple, we didn't have to cut holes in anybody's head. We didn't have to damage any kind of brain. We were able to measure their activity safely, painlessly, fairly, relatively easily. Carl Lashley was an experimental psychologist known for his learning and memory studies in the early to mid 1900s. He purposely destroyed different amounts of cortex in rats and observed how ablation, the term for doing this, how it affected the rat's ability to learn and remember. After damaging their brains, he put the rats in 3 different types of mazes. They differed in complexity. He found that learning didn't seem to be localized in any one part of the brain. All of the rats were able to learn even after having parts of their cortex damaged. He called this equipotentiality, the notion that when one part of the brain is damaged, the surrounding areas compensate for the loss of function. They make up for the injury. His studies provided evidence against the argument of localization. If you remember, one of the reasons that phrenology lost support in the scientific community was because some individuals were able to show that certain functions are in fact localized. Lashley was able to show that learning in general is not as localized as some of the other more specific functions that other researchers had focused on years before. He was focused on learning. He looked at how learning is impacted. And the brain is able to make up for some damage, some injury by allowing areas near the damage to take over that function. He also came up with the principle of mass action and suggested the amount of damage is proportional to the amount of learning, to how well the animal learns. In other words, the more brain tissue he damaged, the more learning was impaired. Donald Hebb was another early neuropsychologist. He wanted to fully integrate physiology and psychology. He is well known for studying the relationship between the brain and behavior. In 1949, Hebb published his book, The Organization of Behavior. In this book, he suggested the learning process occurs through the growth of physical structures within the brain. Learning equals physical brain growth. According to him, thinking occurs through 2 structures: cell assemblies and phase sequences. You can see illustrations of these physical structures here at the bottom of the screen. These illustrations came from his book from 1949. The most basic unit of the nervous system is the cell assembly. It is a set of neurons that have become associated with one another because they've been activated at the same time through repeated experiences over time. So the most basic structure of the nervous system changes as we learn and gain experience exposure. Phase sequences are groups of cell assemblies, groups that once again become connected because they are activated at the same time over and over again. You can see that in the mid-1900s, we were becoming more familiar with how the brain and the nervous system actually work, how they function, and how they relate to behavior. In the last 75 years, we have made even more progress in the field of neuropsychology. I wanted to show you in this section just a high-level overview of two of the most important pioneers in this field and some of the technology that we use to research topics of interest.